So you've seen these terms before. Isotonic means what? What does it mean? It's the same as the surrounding tissue. How about hypotonic? It means that, imagine you're the cell. What's it telling you about the solution around you? Does it have more or less water? Well, I guess it's written up there that way, huh? It has more water around it, right? It's hypotonic. That means what has less? What's it have less of? Okay, just as a refresher, the word tonic, as soon as you see that, you should think solution. And specifically, the solute of the solution. Isotonic means that the solution has exactly the same solute on the outside of the cell as the inside of the cell. Hypotonic means the solution has what outside the cell compared to inside the cell? More or less solute? Hyposolute. What's that telling you? Less solution on the outside, which means if it has less solution, that means it has more water. When you're looking at this concentration, we're not talking about a volume, we're talking about a percentage or a concentration. So if I have a liter of water, that's volume. But when I say that it's 50% water, that's a concentration or percentage. This tonicity concept is talking about the percentage. You have to imagine that it's always going to be a fixed volume, whether it's 5.5 liters of blood or 1 liter of water or whatever it is in your body. When they talk about it being isotonic, it means that that solution is exactly the same as a cell. When it says that it's hypotonic, it means that that solution has less solute, hypo, less, tonic, the solute, than inside the cell. And hypertonic means that the solution around it has hyper, more solute than the cell. And it doesn't, I'm using solute as a general term. It can be salt, it can be sugar. You can actually cure things with sugar. My first week as a sous chef, I actually got to cure meats all week, and it was crazy. We took sugar and salt and poured it all over fish, and we just left it there for a week. And what do you think happened to the fish? Did it start swelling up, or did it shrivel down? It shriveled, because that solution outside the fish's cells was hypertonic, extremely hypertonic. There was no water out there. It was all salt and sugar, so all glucose and sodium and chloride. There was a hypertonic solution. It started pulling all the water out of the fish cells. And why do they do that? They don't do it just to shrink the fish up. They do it to try and do what to the stuff on the surface of the fish? Kill the bacteria. They're trying to suck the water out of that bacteria and kill the bacteria, like when you cure anything. Okay, so now we're going to put a number with this. I know it looks scary when you see 300 millimolar or 300 milliosmol or any of those terms like that. Just imagine they're talking about 300 pieces of whatever the solute is. So if the inside of the cell has 300 pieces of solute and the outside of the cell has 300 pieces of solute, what can you say about that cell or that solution? It's isotonic. So in this situation, if the inside of the cell has 300 pieces and it's hypotonic out here, what's it telling you? It's anything less than 300 pieces. It can be 1 piece. Heck, it can be 0 pieces. What is it if it's 0 pieces of solute? It's a 100% pure water. What's that going to do to your cells if you put them in 100% pure water? Look at the term again. Do you remember when I talked about this before? If you put a cell in a hypotonic solution, it swells up like a gigantic O. If you put it in a hypertonic solution, it shrivels it up because it sucks the water out. So like I said, this is kind of a review, but now we're putting values to it, and I'm going to show you how it works in the kidney. Okay, so first the renal medulla. The renal medulla, is that the outer surface of the kidney, or is it the inner part of the kidney? What's the name for the outer surface of the brain? The cortex. So what do you call the outer surface of the kidney? The renal cortex, right? The renal medulla is down deep inside. The cortex itself is about 300 milliosmoles. Was that on the last slide? No, it wasn't. You might want to jot that somewhere. The cortex, renal cortex, stays at about 300 milliosmoles. So about 300 pieces of solution, or solute, for every volume of solution. But the renal medulla changes. The closer it is to the cortex, it's 300, but the further you get down deeper into the kidney, the more what? Hypo or hypertonic does it become? The number of solute particles rises. It becomes hypertonic, right? So it becomes more hypertonic. What's going to happen to the urine solution down deeper? Will it get more water added to it, or will it get more water sucked out of it when it's going through a hypertonic environment? It gets sucked out. It's pulling the water out of the tubules. Okay, so there are three parts of this that we're going to break down. When you get into the renal medulla and you're talking about the nephron still, the first thing you want to remember is that the medullary countercurrent system is what's doing the work. It's actually doing the work of concentrating urine. It's a system, right? And I would underline the long loops of Henle, because what they do, that is the first part. The loops of Henle establish something called the vertical osmotic gradient. The long loop going way down, this long tube gets more and more hypertonic the further you down. That's what's creating that change in environment. It's creating the change in environment. Next, you want to underline the vasa recta. The vasa recta, all it's doing is preventing the change of that gradient. It's just keeping the gradient consistent with the inside of the tubule as far as it possibly can. It's providing blood, so it's keeping the inside of the kidneys alive. If this gets blocked, your kidney is going to be a goner. If this gets blocked, you can't concentrate urine either. This is like a little stream or river. I'm going to flip ahead real quick. Oh, where did that picture go? I just saw it when we opened this up. There we go. So the vasa recta is just a blood system. As it's going down along the loop of Henle, the tubules, it's just pulling anything that gets sucked out of the tubule is pulling into the vasa recta, and it's like throwing it into a stream. It gets carried downstream really quickly. So we have the long part of the loop of Henle, and then we have the vasa recta, and then where's the third part in here? There we go. The collecting tubules. That's the last one. The collecting tubules are the regulating center. Remember we talked about those macula densa cells? And the collecting tubules and the distal tubule, those are where the collecting center, or the regulating centers are at. So you've got the loop of Henle creating this difference, this gradient. You have the vasa recta that's maintaining everything, and then you've got the very end, the distal tubules and the collecting duct that are regulating everything. And then actually, this says vasopressin, but what other hormone's there? We talked about it on Tuesday. Vasopressin was known as ADH, but there was another hormone we talked about that worked at the distal tubules and the collecting duct to help you retain something else. Not water, but salt. What was it that was helping you retain salt? Aldosterone. Which is another important point if you didn't catch that before. Vasopressin or ADH makes you retain water. Aldosterone makes you retain salt. They don't do both. You can turn on aldosterone and pull in salt, which causes you to want to retain water, but aldosterone doesn't directly affect water. Vasopressin doesn't directly affect salt. So those are the three main components. All right, this is a kidney on its side, just the way the book has it drawn. So right here in the outer cortex, you can see that the milliosmolarity is about 300 milliosmoles, so about 300 parts solute for the solution. And then as you go deeper, like this, come down deeper down along that descending loop of Henle, you get deeper into what's this purpley colored area called? It's the medulla. The further you get down, the more concentrated this is, the higher the amount of solute down in there is, the more hypertonic it is. So it helps pull water out. Remember, you make 180 liters of filtrate every day, but you only pee out 1.5 liters. So as this water is coming down into this hypertonic solution, it's getting the water pulled out, pushed into the bloodstream, and carried back up to the rest of the body. Right? So like I said, those three parts, the first one I mentioned was the loop of Henle. The loop of Henle and the juxtamedullary nephrons actually establish this long gradient. And it's the type of nephron. There are two types. There are cortical and juxtamedullary nephrons. These are the juxtamedullary. What was the difference between the two? Juxtamedullary are very long and they go way down juxta next to the medulla, right? The corticals stay where? Think about it. Cortical nephrons, where are they telling you they're primarily at? The cortex. They just dip their toe, the very tip of the loop, they dip it into the medulla, but that's it. Not much goes into the medulla. These juxtamedullary ones are the ones that do the most concentrating urine. And I give you an example of kangaroo rats, desert animals. They have lots of those juxtamedullary ones. They do a lot of concentrating of urine. Right? And then here are three sections. I kept telling you, you have to know the different parts of the tubule and what happens to the different parts. So here's the proximal tubule. Already on Tuesday, what did I tell you is going on in the proximal tubule? I gave you an example of my sister's purse. It's where most things are, it's dumped out into the Bowman's capsule. It's where most is recollected. So about 80% of everything you dump out, you're going to recollect. 65% of that was sodium, or 67% was sodium. 100% of the sugar, 100% of the amino acids. It's going to try and pull back as much as it possibly can. Right? And then when you're talking about water, about 65% is reabsorbed in the proximal tubule, and roughly 35% is reabsorbed later down the road. Excuse me. All right. Next, the difference between the descending and the ascending limb, obviously one's going down, one's going up. The descending limb is permeable to water, not sodium, and I said this on Tuesday also. So I'm trying to stress the significant things. As it's going down, as that pre-urine's dripping down deep into the medulla, water's getting pulled out, but salt is not getting pulled out. Then you get all the way down to the tip, and as you're coming back up the ascending loop, salt gets pulled out, but what do you know about water? It's not. Those two limbs are exactly the opposite. The one going down pulls out water, the one going up pulls out salt, but they don't do the opposites. Right, so this is an example of how this works. As it starts going down from the cortex into the medulla, it starts at 300 milliosmoles, which is what tonic? Hyper, hypo, or iso? Iso. But then suddenly what you do, and it's actually on this side that the action happens, is you flip on a pump. What pump do you instinctively want to say gets flipped on? Sodium-potassium pumps, right? So it flips on a sodium pump here, and it's pulling in sodium, and the chloride wants to follow it. As it pulls in sodium, it lowers the tonicity on this side. So it's becoming a little bit more hypotonic. But what it's doing to the tissue in between those two tubules, remember this tubule is this long thing, and there's not air in between. There's actually tissue. Lots of kidney cells inside of there. So it's pumping all of this salt in here, and what's it doing to this tissue? It's making it hypertonic. So can it pull water out of this side where it's pulling the salt? No, this is the ascending side. It's only permeable to salt. So you're pulling the salt or pumping the salt out on this side, which is creating this hypertonic solution. And now suddenly water starts getting pulled out of the descending side as a result. This is a passive process. What do you call that passive movement of water? Osmosis. So it's pulling it out. And what's interesting is the more salt you pump, the more hypertonic it becomes. You pull more water out. The more salt you pump, more hypertonic it becomes. And it just keeps doing this so that until you get all the way down the tip, it becomes so hypertonic down here, you're at about roughly 4 times the tonicity. About 4 times as hypertonic as you are in the cortex. So it's this long loop of handling these pumps over here that are actually creating this gradient. They do the creation. What actually maintains the osmolarity? What's the name of the structure that goes right along with this and maintains the osmolarity? It was the blood vessel. What was the name of the blood vessel? It started with a V. Vasa recta. So the vasa recta goes right along it, and they call this the countercurrent exchange, not countercurrent multiplication. It's the countercurrent exchange because as this thing flows along, it stays at the same tonicity as whatever the tissue around it is. So if the tissue is 300 milliosmol up here, it's going to stay exactly the same as that tissue. If it's 1200 way down here deep in the medulla, it's going to stay exactly the same as the tissue. All this is doing is it's acting like a stream, a fast-flowing stream. Blood flows along here, pulls out the water or salt or whatever's in the environment, and then pulls it off to the rest of the body. It doesn't stop to do a lot of swapping. It just pulls in whatever you need and carries it off to the rest of the body. It's there for maintenance, and that's it. And you think that's all this says here. Yeah, it prevents dissolution, and it maintains everything. It equilibrates with whatever environment it's around. So if I were to ask you something about this, which is true about the osmotic gradient? Which is the true statement about the osmotic gradient? How about number 1? The loop of Henle is permeable to water on the descending limb. False. It's permeable to water on what side? Ascending. What's the descending limb permeable to? Oh, what am I talking about? That is right. Sorry, in my mind I was thinking salt. So that one was pretty true. I would have missed that one on the test, because I would have just jumped to conclusions. Anyway, so hopefully the other ones are false. How about number 2? The cortex has an osmotic gradient that goes from 300 to 1200. Why is it false? That's the medulla, right? The cortex stays at 300. How about number 3? The vasorecta created the osmotic gradient. Excuse me. The vasorecta created the osmotic gradient. What created the osmotic gradient? The long loop of Henle. What's the vasorecta do? It just maintains it. Number 4, the name associated with the vasorecta is the countercurrent multiplication system. It's the countercurrent exchange. Yeah, it's just a swap. So number 1 was the true answer. Okay, I would have been really confused on the test because I would have crossed all 4 of them out and went, uh-oh. Anyway, next one, aldosterone. Again, this is like the 5th time you've seen aldosterone on a slide, so you probably have to know it. In fact, if I've said it 5 times now, count on a test, and not just this test, but the final exam too, which is not cumulative, don't freak out. But the final exam's all hormones, so aldosterone will be back. All right, where it works, the distal tubule and the collecting duct, you can add the collecting duct. Some textbooks go back and forth, and some say it's just distal tubule, other the collecting duct, but most of the books I've read or taught out of say both do it. So just on the road in case like an NCLEX or MCAT hits you with that, you know. All right, stimulates absorption of sodium and secretion of potassium. Why? What's it turn on? Sodium-potassium pump. Aldosterone directly turns on the sodium-potassium pump. So it comes down here to the distal tubule and collecting duct. It goes specifically to these cells. Binds to what's it have to bind to? It's a hormone. It has to bind to a receptor. Binds to a receptor and turns on a machine that opens up the sodium-potassium pump. This is showing vasopressin, not aldosterone. So it's the same idea. It binds to a receptor that turns on that little sodium-potassium pump and starts cranking sodium over. Right? So there it is. And of course you know, if sodium goes one way, what happens to potassium? It goes the opposite. And by the way, sodium always goes into the blood or out to the tubule. Sodium is always being pulled into the blood. It's never being pumped out to the tubule, which means potassium is always being pumped out to the tubule by the sodium-potassium pump. Okay, vasopressin, another hormone. So you'll have to know now for this exam and for the last exam. It's also known as ADH, or antidiuretic hormone. This is a fun hormone. This is the one that, what is today, Thursday? If you don't have class tomorrow, you'll probably be out there testing your antidiuretic hormone tonight. Because when you drink alcohol, it affects ADH. So vasopressin works by helping you retain water. Vasopressin makes you retain water. Without vasopressin, you just pee out the water. It's called an antidiuretic. So a diuretic makes you pee, an antidiuretic makes you retain. Exactly. Where this is released from is the hypothalamus. It's right there. So it's made and released from the hypothalamus. And of course, you know alcohol directly affects the brain, so we'll build up to that. It works at the distal tubule and collecting duct to help you retain water. Why would you do this, by the way? Why would you want to retain water? What could be happening to your blood? Maybe your blood volume is too low. Maybe your blood pressure is too low. Maybe you have too much what in your blood already? Salt. Yeah. Thanks for leading into that. So yeah, maybe you have too much salt. If you have too much solute in your body, you'll start retaining water. Think about it. When you eat a bunch of pretzels, what happens? You drink water, you retain a lot of water. You don't pee it out right away. You drink that water and you feel bloaty later, right? So those are some of the reasons that ADH is released. So stimuli because of hypertonicity, in other words, too much solute in your blood. Hypotension, what's that mean? Low blood pressure, yep. And those hypothalamic osmoreceptors that are detecting concentration and baroreceptors that are detecting blood pressure get turned on. So when you go out and you start drinking, after the first couple beers, that alcohol goes to your head, and you know that. It affects your frontal lobe and your personality, goes a little deeper to the hypothalamus. Think about it. Hypothalamus, what were the 4 things? Freezing, yeah, S, right? Freezing, fighting, right, freezing, feeding, fighting, and fornicating. Think about it. When you drink alcohol, all 4 of those are affected. What about the temperature? When you start drinking alcohol, how do you start feeling? You're like hot. You're like, ugh. When you start taking your coat off or whatever, even though it's freezing outside, you start drinking, right? You get the munchies, so there's your feeding, and then, of course, your beer goggles slide on, and may lead to the last F. But all those things change because of this. One thing that happens is that you turn off antidiuretic hormone, which means what starts happening? If you turn off an antidiuretic, you start falling into what state? Turn off an antidiuretic. What's the opposite of antidiuretic? Diuretic. Yeah, you go into a diuretic state, and the next thing you know, you broke the seal and you're peeing every 5 minutes, right? And you actually pee out a lot of water. There's not a lot of salt in that, so you retain a lot of salt, but you lose a lot of water. How do you feel the next morning? You feel hypertonic, like your body is dehydrated. And that's what happened. Right? And then here's how vasopressin works. So vasopressin binds to a receptor, turns on some machinery on the inside that opens up little pores. When I was learning this, I always thought, well, it's always digging holes for water, because it puts these little holes in here and lets water seep into your blood. You don't have it, it blocks the holes, and the water leaves your body. Pee it out. ADH, antidiuretic, always digging holes. So here's an example. Of course, you know that excuse me, water's not permeable here, going up. But once it gets into the end of the distal tubule and the collecting ducts, you can change this. If your urine's too dilute, what are you going to do with ADH? Are you going to turn it on or turn it off? If your urine's too watery, what are you going to do with antidiuretic hormone? You turn it on, which makes you stop losing the water, right? And you start retaining it. So without ADH, you actually block all of this, and you urinate out whatever you created down here. So when you go drinking, what color does your urine turn when you can't release ADH? It turns extremely clear and colorless because you're just releasing lots of dilute watery urine. If you made too much ADH, what color would your pee be? Dark yellow, right? Because you're concentrating, you're pulling all the water you possibly can in. It's interesting, vasopressin is actually, oxytocin is released after an orgasm for a woman, vasopressin is released after an orgasm for a man. It's kind of a weird thing. So we actually retain water after sex for some reason. What's also interesting about vasopressin is it increases memory, which doesn't explain why we never remember your name after sex. Kidding. But anyway, vasopressin comes out. Like desmopressin. I've heard of some doctors that prescribe desmopressin for memory issues because it helps increase memory. The bad thing is that it also makes you retain lots and lots of water, so it raises your blood pressure. It causes problems there too. And I'll flip before anybody wants to crucify me for making a joke like that. So which is true about vasopressin? How about number 1? Vasopressin is a hormone made in the kidneys. No. It works on the kidneys, but it's made where? Hypothalamus. Number 2, vasopressin stimulates the reabsorption of sodium. No. Vasopressin stimulates the reabsorption of water. What causes reabsorption of sodium? Aldosterone. How about number 3? Vasopressin has another name, which is aldosterone. What's its other name? ADH, antidiuretic hormone. How about number 4? Vasopressin binds to receptors in the distal tubule and collecting duct. True. Yep, it binds those receptors back here and turns those little holes on. Okay. So the tubular segments that are permeable to water, excuse me. The areas that are permeable to water, that wants to chase the solute. What's the solute usually? Salt, sodium chloride, right? It can be sugars too. For instance, when diabetics, this is a really good example, diabetics can't get all the sugar back into their blood, so they have a lot of sugar in their filtrate. What's that going to do to water? Will it push the water back into the blood, or will it keep the water in this filtrate? It keeps the water in the filtrate, so these people that are diabetic and have lots of sugar in their urine, what else do they have a problem with? They lose a lot of water. That's why they have polyuria and they're peeing all the time. It's because they're losing lots of water, which makes them what? Dehydrated, which makes them thirsty, which means they always want to eat too. Try and bring that water and that sugar back in. Okay, so these are just kind of keys. Solute excretion is always accompanied by comparable water excretion because of osmosis. So when you secrete the solutes, the water chases it. When you bring back the solutes, the water chases it. There are two types of diuresis. There's osmotic diuresis, which means the concentration is consistent with normal urine. So what color would that urine be? It would be yellow. It would be like normal urine. It's going to have the right concentrations. So in other words, you're losing lots of salt, lots of urea, lots of urobilinogen. You're losing all of those things. Water diuresis means you're primarily losing water. So what color is your urine going to be? Clear. What does alcohol do? Osmotic or water diuresis? Water diuresis. Yep. Okay, and then renal failure. You can live. Can you live with one kidney at all? Yeah, you can live with one kidney. When both of your kidneys have actually lost 70% of their function, then you go into renal failure. So your kidneys can actually take a lot of loss. In fact, every day you're losing more and more nephrons. Can you grow them back if you eat the right diet? Nope. Once they're gone, they're always gone. So it's better just to take care of it. Drink plenty of fluids and keep them clean. Because even when you get little kidney stones and they damage the nephrons or damage walls of the kidney, those nephrons can't come back. Okay, and same with taking drugs. When you take a lot of Tylenol or a lot of aspirin, it has a huge impact on your kidney. Or other things like penicillin can do that too. So anyway, occurs when the body can't keep up with the renal demands. So here are some causes. Infectious organisms. They can come from two directions. They can come from the outside in, or they can come from the blood into the kidney. More commonly, it actually comes from the blood to the kidney when you have an infectious organism. So like a bacterial infection in your blood that gets down in your kidneys and causes problems. Toxic agents, that would be like I just mentioned earlier, things like Tylenol, aspirin, ibuprofen, drugs that you take too much of. The kidney's going to try and clear those, and if they're protein-based or they're even small peptides, they can get clogged up in the filters and cause things that are called glomerulonephritis. Think about that word. Glomerulo is referring to what? Glomerulus, neph is the nephron, and itis is telling you inflammation. So I love words like that. If you're into anatomy and physiology, if you look at the words, it kind of tells you what the disease is. Right? Inappropriate immune response, actually another glomerulonephritis, but think about antibodies. They're little pieces of protein. If your body's making way too many antibodies, what could happen? They could go to the kidney and do what? Imagine your spaghetti strainers in the kidney. When you take your spaghetti, you cook rice in the same container with the spaghetti and you just dump it all in. What's going to happen? The rice is going to plug up all the holes, and what's going to happen? You're not going to be able to drain that spaghetti water. That's what happens with the immune system. When it's overreactive, things like lupus can do this. Makes lots of antibodies, and they go and they stick inside the kidneys, and they cause problems with the kidneys. Obstruction of urine flow, what could do that? A kidney stone, tumors near the kidney. In fact, it doesn't even have to be in the kidney. It could be compressing the ureter, right? It could be compressing urethra. What sex has a problem with that happening? Males, because of what organ? Prostate, yep. So obstructed urine flow backs all the urine up, and then it starts increasing pressure on the kidney. The kidney enlarges because of prostate issues. And it actually starts destroying the kidney. And then insufficient renal blood supply, things like shock can do that. So if you decrease the blood supply to the kidneys, because you're trying to get it to the brain, the kidneys are sensitive and they'll actually start shutting down. Liver and kidneys are usually the first organs to shut down in renal failure. Alright, and then the next thing, urine. So urine's temporarily stored in the bladder. What are the name of the tubes that carry it from the kidney to the bladder? The ureters. Yep. And the ureters actually work by micturition. I always looked at them and I thought they must be like a drip, like when you have an IV bag hanging and it drips down, but it's not. Those ureters are constantly squeezing, just like your esophagus squeezes food. It's constantly squeezing it down by peristalsis. So it's kind of like it's milking the urine out of the kidneys. All right, this works so much like defecation, it's crazy. So in defecation, the internal anal sphincter was made of what type of muscle? The internal is smooth, just like this. The external was skeletal. It's the same type of reflex too. The bladder stretches. When the stretch receptors detect them, they say that you've got about 300 to 500 milliliters of fluid in there. So about a half liter of fluid is all it takes. It stretches out and sends a signal to the spinal cord. The signal comes back and tells the internal urethral sphincter to relax. Luckily, if your spinal cord's intact, your brain sends a signal down and tells the external urethral sphincter to contract until you're at the appropriate place. What do they call it if you don't have that connection, or that connection's broken, or any of the neuron pathways, or even those sphincters are broken? What disorder do you have? Incontinence. Yep. And there are lots of types of incontinence. There's overflow incontinence where you filled your bladder too much and then you have a good laugh. Oops. There are other things that can be caused by STDs. STDs are that close already, they just go straight in and start affecting that neural pathway. Like syphilis. Syphilis will get in, go to the spinal cord, just destroy that area of the spinal cord. All right, and then, it's right down there, urinary incontinence. And then even with babies, they have incontinence, but it's because their spinal cord hasn't developed yet. They just can't control it. So this is the same pathway we described when we talked about the defecation response. You can see the bladder fills, stretch receptors are turned on, parasympathetic nerve pathways turned on as a reflex. The bladder starts squeezing, the internal internal urethral sphincter starts relaxing. But luckily, as long as the brain's involved consciously, you send a signal down to shut off that response so you don't wet your pants until it's appropriate. Well, it's never really appropriate, but you know what I mean. And then here it shows the stretch response. You can start accumulating urine, you keep accumulating more, but there's really not a lot of pressure until you get about 300 to 500 milliliters, and at this point, you have to pee. It's time to pull the car over. And that's everything for the kidney. This next set is a ton of review. There's really only 3 new sections in it. I may as well stop and